Hi, everybody. It's me and my cat, Mac. He's going to be 18 this year. He's a old boy. Okay, so we're in my living room now, and we're going to talk about a very complicated topic that's very close to my heart. I did a lot of research on this stuff when I was an active researcher. So we're going to talk about some of the mechanisms by which transcription factors do their job. We're going to talk about chromatin remodeling, histone variants, histone modifications, and some other ways that transcription factors function. So remember where we left off, right? You have proteins, transcription factors that can bind to DNA in a sequence-specific manner and influence transcription. You get these transcription factors that are called regulatory transcription factors that are specific for certain genes, and general transcription factors that are required at every gene. And these transcription factors bind to DNA at enhancers. If they're going to upregulate a gene, those are activators that bind to enhancers. And then there are inhibitors or repressors that bind to silencers that turn transcription down. So notice that if you look at some of these enhancers, you could also, silencers work in the same way. You notice that they are sometimes far from the promoter of the gene that they regulate. And interestingly, most of these guys don't actually talk or rather bind to RNA polymerase directly. There's different ways that transcription factors can talk to RNA polymerase. The first two are through basically general transcription factors that are always there to help genes be transcribed. Transcription factor 2D is a very famous complex of proteins, as is the mediator complex. And we're going to talk about how transcription factors work through those guys to begin with. And then after we've talked about transcription factors a little bit more, we'll talk about nucleosome composition and chromatin remodeling. But we'll deal with the first two first. So the first two are relatively simple. So the first two mechanisms by which transcription factors can affect gene activity are through two very famous complexes of proteins. One is called TF2D. So TF2D, or transcription factor 2D, to give it its full name, is really important for recruiting RNA polymerase to the core promoter of genes. So we haven't talked about this too much yet, but it is true that RNA polymerase, which, as you know, is recruited to the core promoter to transcribe a gene, it needs help. One of the things that helps it is a complex called TF2D. And because of that, transcription factors can regulate the activity of TF2D. And so what is shown here is a way that an activator protein can bind to an enhancer and regulate TF2D, and in turn regulate RNA polymerase. And so what you see here is there is an activator protein, transcription factor, it's bound to this enhancer. It's talking to TF2D through another co-activator protein. And TF2D is bound to the core promoter. Now in this case, this gene is going to be on, transcription will occur because TF2D is required to get RNA polymerase 2 to do its job. Conversely, here is an example of where a repressor protein is binding to a silencer protein, silencer sequence. And look at where TF2D is in the second case. It is not at the core promoter. The repressor protein is kind of holding it off the core promoter. In that case, this gene would be silenced and not transcribed. So these are two ways that different types of transcription factors, either activating transcription factors or repressing transcription factors, can regulate transcription. Not through direct interaction with RNA polymerase, rather through RNA polymerase's best friend, TF2D. Another complex of proteins, TF2D is always written as like one blob, but it's actually a lot of different proteins. And its job is to help RNA polymerase work. The second thing that we're going to talk about, this mediated complex, is conceptually pretty much identical. So what you're seeing here is a picture of the mediator complex. Here it is, it's this big, I don't know, crazy looking red blob. The mediator complex, as well as TF2D, are required to get RNA polymerase to do its job. The mediator actually does something very specific. What the mediator does is help phosphorylate RNA polymerase. So there's little phosphate groups right here. You can see this PO4 thing. That's a phosphate group. The mediator helps tag a phosphate onto RNA polymerase because that is required for RNA polymerase to transcribe a gene. So an enhancer sequence can recruit an activating transcription factor, and its job is to help the mediator form this big, stable complex with transcription factor 2D, RNA polymerase, other general transcription factors, as shown here. And this whole massive bunch of proteins is required for transcription to occur. If the enhancer doesn't bind the activator protein, it won't recruit the mediator, RNA polymerase will not be phosphorylated, and you won't get transcription. So when the enhancer binds the activator and helps this big load of proteins get together, RNA polymerase does its job. Again, right, this is conceptually, some of the names of the proteins are different, but conceptually, it's just the same as this. Activating transcription factors are helping form this complex that is going to transcribe a gene. It should then not really come as a shock that if there's a silencer DNA sequence that is recruiting a repressor protein, basically RNA polymerase is not phosphorylated. So in this case, the repressor protein talks to the mediator and tells it, don't phosphorylate RNA polymerase, and then the gene will not be transcribed. So in both of those cases, both the TF2D case and the mediated case, activators make the complex work, repressors stop the complex working. It's kind of complicated when you think about the details, but the big picture, you know, an activating transcription factor causes a stable complex that favors transcription to occur, and the opposite is true with a repressing transcription factor. I'm going to take a quick aside to talk about how regulatory transcription factors can be affected by small molecules and other ways. So while we're talking about transcription factors and what they can do, it's a good time to talk about this. So just like the lac repressor, if you remember the lac repressor, right? It's this protein that binds to a DNA sequence. When it binds to allolactose, it falls off. That binding caused a three-dimensional shape change in the repressor, which causes it to drop off the DNA. Eukaryotic transcription factors can also be regulated in a pretty similar way. And I'm going to just discuss three of the more common ways they can be regulated. The first one is binding a small molecule, just like allolactose. The second one is you can get transcription factors that stick to each other in a protein-protein interaction to form a functional transcription factor unit. And then there's covalent modification of transcription factors. And all that means is a transcription factor has something covalently bonded to it, like a phosphate group. It's a small, chemical moiety that's bound to the protein by a covalent bond. So here's one example. This is how hormone receptors work. So here's an example of a hormone. It's binding to this transcription factor. Without the hormone being bound, this activating factor will not bind to the response element. As soon as this green hormone binds to it, however, it will cause a shape change in the protein, which will allow it to bind DNA. And that can cause the transcription factor to now do its job. Here's an example of protein-protein interactions. Here we have two transcription factors that are required to talk to each other via a protein-protein interaction before they can bind DNA. So this could be two of the same thing, that's a homodimer, or it could be two different proteins, which would be a heterodimer. But in any case, it requires two proteins to join together to bind DNA. The third case is a covalent modification. So in this case, these transcription factors, this happens to be a dimer. It's bound to DNA, but it's inactive until it becomes covalently modified. In this case, until it's phosphorylated. These are three general ways that transcription factors can be regulated. And I'm going to show you a couple of examples of those in real life situations. So steroid hormones, right? We all know what steroid hormones are. They're things like testosterone and estrogen. There's lots and lots more. These are relatively small molecules that are released by things called endocrine glands in your body. And then they are released into your blood. And then essentially what they do is they go all over your body and they actually get into basically every cell in your body because steroid hormones can cross biological membranes and they just diffuse throughout your whole body. However, not every single cell in your body will respond when it sees a hormone and a hormone gets into that cell. And that is because in order for a cell to respond to that hormone, it needs a receptor for that hormone. Now, steroid hormone receptors are kind of interesting because they're just proteins and they're actually transcription factors and also receptors in one. So the binding of the steroid hormone to the transcription factor is what turns it on. So that's kind of like a small molecule binding to a protein. So here we have an example of that. This is a real example that happens in your body. Here's a hormone. This one's called glucocorticoid. It's a pretty small molecule. It can diffuse across the membrane, but it won't do anything in a cell unless the glucocorticoid receptor is present. So this shows a cell in which that is true. So here is a glucocorticoid receptor, and the mechanism of action is pretty neat. So here's what happens. The glucocorticoid receptor is sitting around in the cytoplasm of this cell, bound by these two different proteins. They're both the two examples of the same protein, HSP90. Now, when the glucocorticoid receptor is bound by the hormone, these HSP proteins, actually fall off. And when they fall off, they actually cause a dimerization of two glucocorticoid receptors. When this happens, so here we have now a dimer that's been formed, this guy moves into the nucleus and actually acts like a transcription factor. So here it is binding a specific DNA sequence next to the core promoter of a target gene for this hormone, and it turns the gene on. Now, in this case, this response element, which is effectively an enhancer, has got a special name. It's called a glucocorticoid response element. And it's kind of cool. These GREs, these glucocorticoid response elements, are located all over your genome next to genes that need to be turned on by this hormone. And so this is a really cool way to do it, right? glucocorticoid binds to this receptor, causing it to dimerize, move into the nucleus, where it binds wherever it finds its specific sequence and turns on a gene. So it can activate many genes very quickly at the same time. So that's one example in the real world of how small molecule binding can cause one of these transcription factors to activate. It's kind of cool because it talks about a couple of the different principles we've already talked about: small molecule binding and dimerization. Here's another example of that. This is from the CREB Protein Transcription Factor. This is an example of covalent modification now, and how that can affect a transcription factor. The CREB protein is a kind of a famous regulatory transcription factor. It's an acronym for CAMP response element binding protein. And basically, this is a protein that sits around in your cells, and it will only become activated when the cell that it's sitting in is signaled to by a special signaling molecule. Signaling molecules, kind of like hormones, they can move throughout your body, but they can't cross membranes. So they have to signal through the membrane in some way. Now, in the case of the CREB protein, if a cell responds to a special signaling molecule, levels of CAMP, which is cyclic adenosine monophosphate, a very famous signaling molecule that's inside a cell, will go up. When it goes up, then the CREB protein recognizes a CREB response element, kind of like the glucocorticoid response element, and activates genes. So again, the details here are not important for you guys to understand. They're just kind of interesting. But I want to give you an example of covalent modification and how it can change a transcription factor. So here we have an extracellular signaling molecule in purple. It's going to bind to a receptor, this time on the surface of a cell. There's a whole chain of things that happen inside the cell, but the upshot is that you get an increase in this gray molecule, CAMP. This gray molecule activates a protein kinase. Its job is to tack on phosphate groups to proteins, and pKa, this protein kinase A molecule, moves into the nucleus, tags these phosphate groups onto the CREB protein, which can then turn on a target gene. So this is an example of covalent modification activating a transcription factor. Again, this is a real world version of this concept. So look at those last two examples as examples of these mechanisms of action of transcription factors. So it kind of puts those concepts into real world practice. All right, the last thing I want to talk about in this little lecture is chromatin remodeling. So transcription factors can work in multiple ways, as you guys now are understanding, right? Through TF2D, through the mediator. Their activity is affected by small molecules, covalent modifications, dimerization. And finally, another thing they can do is to change the composition of chromatin in your cells. So just a quick reminder of what chromatin actually is. Chromatin is the packaging of your DNA in your nucleus. So your DNA needs to be compacted a lot to fit into your nucleus. And chromatin is the name of the DNA and protein complex that is sort of scrunched together in your nucleus. When the chromatin is compact, it's said that it has a closed conformation. And this makes transcription very difficult because it's sort of like imagining a ball of string that's packed together. And then RNA polymerase would have to get in there to find the gene that it needs to turn on. It's hard to do. An open conformation is when the chromatin is of a looser structure And that means it's accessible for transcription factors and RNA polymerase to get in there and transcribe genes. So here's what it looks like in your cells. Here's some DNA, the classic double helix. There's a diagram of it in blue, and there's also an electron micrograph of it above. DNA is wound around these things called nucleosomes. Here's an example of DNA wound around a nucleosome. It's actually wound around them twice. Nucleosomes are really important, so I want to just talk about them for 1 second. A nucleosome is a little ball of proteins. It's actually an octamer. It's made of 8 proteins, and they're all histones. So there's 8 different proteins that are called histones. They're shown up here. They kind of look like tadpoles because they've got a tail, which is going to be important in a minute. And each nucleosome is consisting of 8 of those things. So here's DNA wrapped around these histones, these nucleosomes twice. And then the nucleosomes can actually wrap around each other to give what is called a 30 nanometer fiber, another compacted structure. And then finally, this 30 nanometer fiber can also be sort of compacted into these 300 nanometer fibers. So you can see how it's very compacted, right? And then finally, when your DNA is at its sort of most obviously compacted is at metaphase. And so this shows you a metaphase chromosome, which is what you guys know and love from Mendelian genetics. So your DNA, bottom line, your DNA is wrapped around into a structure called chromatin that makes it very hard to transcribe genes. So we need to loosen it up a bit if we want to transcribe a gene. And transcription factors can recruit proteins that are going to do that loosening of this structure. And we're going to talk about histone tails specifically in a minute as one way of the way that they do that. So we're going to focus in kind of on these nucleosomes and the DNA that's wrapped around them and how that can change when a gene needs to be transcribed. So this is a process called ATP-dependent chromatin remodeling. And it's called that because it's an energy-dependent process. So ATP is required to basically make DNA more accessible so genes can be transcribed. So at the top of this diagram, we see a highly compacted piece of DNA with these nucleosomes wrapped around, DNA wrapped around the nucleosomes that are compacted together. One thing that can happen to loosen these guys up a bit is to change the relative position of a few nucleosomes, as you can see on the left, or you can spread them out, as you can see on the right. This is one aspect of chromatin remodeling. The idea is that we're loosening up the chromatin to allow RNA polymerase to get in there, transcribe a gene. Eukaryotes actually have a lot of protein families that can do this, that are called chromatin remodelers. A couple of the more common ones or more famous ones are shown in the box. And these guys, using the power of ATP, are going to spread the nucleosomes around to make them more make the DNA more accessible to RNA polymerase. Sorry, my cat's looking at me like I'm insane. How do transcription factors fit into this process? Well, they actually recruit or bring in chromatin remodelers to genes to expand the chromatin at those genes. Other ways that chromatin structure can be loosened are things like removing histone octamers, aka nucleosomes, these balls. You can remove those from a piece of DNA to open it up. This creates gaps with no nucleosomes to allow other proteins to come and bind to genes. Or you could actually recruit variant histones, which is relatively new discovery. So the human genome doesn't actually just contain one histone protein. It contains over 70 of them. It's a gene family, which began by gene duplication, as you guys know. And just like other gene families, all these histones accumulated mutations, which gives them slightly different properties, some of which allow genes to be more easily transcribed. Lastly, here are examples of the 4 most common histones that are used to make nucleosomes. And there's something called a histone code, which is this. Transcription factors will recruit enzymes that will chemically modify histones by tacking things like acetyl groups, as shown here, this little AC, or phosphate groups, this little P, or methyl groups, these little M's. And they will chemically modify the tails of these histones in a very specific way that's now called the histone code. And if histones get tagged by these certain modifications, that can recruit more proteins into the gene, depending on exactly the modifications that have occurred, to change the transcription of a gene. Okay, we're almost there. Bear with me. You may be wondering, well, hey, Dr. Parker, if these genes are buried in the chromatin, how do you even start this process? How does a transcription factor get in to recruit things like the mediator complex or a chromatin remodeling complex or something else? Well, that's because there's actually a structure to the way that the DNA is packaged in your nucleus. And it's often true that there's a nucleosome-free region at the beginning and the end of a gene. And that leaves this little gap in the chromatin structure, which allows proteins to move in and bind to the core promoter and regulate transcription of a gene. So what I want to end this little lecture with is talking about how a gene is activated in a eukaryote. So let's begin at the beginning. We have a gene that is basically here in the middle of this bunch of histones. As I just mentioned, there is a nucleosome-free region at each end of that coding region, each end of that gene. If we get to the business end here, we see that there's a core promoter and an enhancer. You notice the enhancer is kind of out in the open. It's not buried in the chromatin. That allows a transcription factor to bind to it. So this green protein is bound to this enhancer. You notice that it's bound to a small molecule, so maybe there was like a trigger that allowed that to happen. So the activator binds to the enhancer. What happens next? Well, the activator can recruit a chromatin remodeler, which does something like acetylates the histone tails, the nearby histones, in an ATP-dependent manner. This allows the transcription pre-initiation complex to fall, which is RNA polymerase plus TF2D plus a bunch of other proteins. And that allows transcription initiation to occur. And then RNA polymerase will start to move down this gene to transcribe it into mRNA. While it's moving, you might wonder what's going to happen to these histones. And one of the things that's known to happen is basically histones jump over RNA polymerase as it moves, as it travels down this gene. And here it goes, making its RNA. Once the RNA is made, of course, it'll be translated into DNA and, translated into protein, and then the gene expression process will be complete. I understand what I just said is a lot of detail. So you might have to listen to this lecture a couple times, but think of the general concepts that we're talking about. In each case, when you're talking about gene activation, you get a transcription factor that binds to an enhancer, a specific DNA sequence, and then That's a trigger that's going to allow tons of other proteins to be recruited to the core promoter, which allows transcription of a gene. The details are different with each one of the mechanisms, but the overall concept is the same. All right, everybody. The last lecture is going to be a lot shorter and a lot simpler, so thank goodness for that. So it's goodbye from me, and it's goodbye from Max. So take it easy, you guys.